Hello, biology superstars! Welcome to the first video for the B1.1 content. This is all about carbohydrates and lipids. So theme B is form and function, and this is the first form and function video that I'm making. I really like this as a theme in the IB biology content because I think in biology in general for a long time, this has been like an organizing idea of biological sciences where form is interwoven with function. So that's when you're looking at this information about the theme. It says biology is the study of life in life. The structure or form of a given molecule or cell or organism or ecosystem is deeply interwoven with its purpose or function. And at the molecular level, we will look at carbohydrates and lipids as evidence of that. And I love carbohydrates and lipids for this because, as it says here, they are carbon-based biomolecules essential for life, and these molecules can form in many different ways, and this deeply impacts their function. So I really do think these two types of molecules are such a strong example in this form and function theme. Guiding and linking questions. Guiding questions we have, hold on, I'm gonna get my pen. I think I'm gonna go blue. So in what ways do variations in form allow diversity of function in both carbohydrates and lipids? And you can compare those two things a little bit. And then also, how do carbohydrates and lipids compare as energy storage compounds? And we'll talk about the different ways they store energy and the different kind of molecules they form quite a lot. Linking questions, how can compounds synthesized by living organisms accumulate and become carbon sinks? So we're looking at carbon-based biomolecules here. In something like C4.2, we look at a whole ecosystem as a carbon sink or a carbon source, so there's a linkage there. And the second linking question is, what are the roles of oxidation and reduction in biological systems? So oxidation and reduction, or redox reactions, where something is oxidized and something else is reduced. Something loses electrons, and in order to do that, something else needs to gain electrons. That all comes up significantly more in the HL content in biology. Like, for example, in C1.2.7, we look at oxidation specifically in cell respiration. It doesn't really come up that much in the SL stuff, but I do think it's useful to think about like the roles of chemical processes as energy transfers in biological systems, no matter what level you're studying at. All right, so let's get looking at our associated skill. And here I'm putting the associated skill as physical and digital molecular modeling. Because when you're looking at the form and function of different types of molecules, it is important to be able to draw out their form in order to discuss their function. So the drawing that we have here, I'm going to see if I can get my pen going with an actual pen. There we go. The drawing that we have here is a carbohydrate, specifically two isomers of glucose. This is alpha glucose over here that has the OH group at the bottom of the first carbon. This is beta glucose over here that has an annoyingly, like the formatting isn't so good, but you can see the OH group at the top of this first carbon. And the drawing of this is not a required skill in the IB curriculum anymore, but just thinking about how this small difference in form can cause a change in function, I think is like important when you think about molecular modeling. So here you have a small change in form that leads to change in function. And the same thing is true with lipid molecules. Two of the lipid molecules we'll look at in some depth in this section of the syllabus are triglycerides and phospholipids. Both of those have a glycerol and a triglyceride. There's 3 fatty acid tails attached to the glycerol. In a phospholipid, there's 1 phosphate group and 2 fatty acid tails. attached to the glycerol. So again, that like small, or in this case, it's not even that small of a difference in form, but the difference in form leads to these molecules having very different functions. So I think there's a lot of places in the IB syllabus where the idea of just even like quickly sketching out a molecular model can be meaningful for your understanding of the content. All right, and I think that is it for our theme and questions and associated skills. The next video, we'll start diving into the content.